Yeah, maybe we'll just nail the water. You could be crying and writing checks.
Not gonna do all of that. So,
can we just set the presentation?
yeah.
That's a good
Let's start, and we'll remove the lights there. You can remove them. So eat your pizza in the door. Let me introduce you, Paul Salma.
Sorry.
Sorry. Who's coming from bioinformatics background, and he actually worked for a while at a non-profit, so should understand how we function here. And now he's a businessman who started a company, founder and a CEO actually at two companies, ElectroSeq and aquaticus, for sequencing. And the invention, it's based on the invention of a fast and cheap device to sequence our genome. So I guess that's what we will hear about. From what I heard, it's like 23andMe, but cheaper and faster and more encompassing. Let's see if if I got it right.
Great, thank you. So this is Angelina Jolie, and she has a family history of breast cancer, and she was genetically tested and found to carry a risk variant that gives her a 90% chance of developing breast cancer in her lifetime. So she responded to that information by having a prophylactic mastectomy and reconstruction, and she's gone from having a 90% risk of having cancer to a 0% risk. And she's going to live to hold her grandchildren. So she is one example of what we are now calling precision medicine, using genomics as part of precision medicine to tailor the treatment to the patient's genetic variation. So it's not one size fits all anymore. It's tailoring medicine to precisely fit a patient. So I'm Paul Szauter, CEO and founder of ElectroSeq, and we are improving DNA sequencing technology to drive precision medicine. So there's three areas that I'd like to talk about today. The first is precision medicine, which I just introduced. The second is technological change, because it's impossible to talk about DNA sequencing without dealing with technological change. And I'd like to generalize this by looking at the development of technology in information science, information processing. And finally, we'll talk about some of the details of DNA sequencing and how we're improving it. So let's start with precision medicine. What is precision medicine? Well, it's going to be kind of a big data problem, so there's more to it than genomics. I'm focused on the genomics aspect of it. But how many people here have had their genome sequenced? Oh, cool. All right. Usually I don't see any hands. I was part of the Personal Genome Project, so I had my sequence done in 2012. But most hands don't go up here, and that's why we need a precision medicine initiative. So this is President Obama announcing the Precision Medicine Initiative in January of 2015. And I have to tell you, anytime you see the President of the United States with a DNA model in the White House press room, you know it's going to be a good day for genomics. So the slide in his background is one of the logos for the Precision Medicine Initiative. This is an NIH-wide initiative to improve the delivery of medical care and ultimately lower the cost. So is anyone here involved in any aspect of precision medicine? I think that would be a component to something that somebody's doing here. Not yet? Okay, but you will be. So the concept of precision medicine is not really a new one. In genomics, what we'd like to do is identify people early, because the problem with medical care, one of the problems with medical care, the reason it costs so much, is we have a model where we come in late when people are at end stages of disease and we do heroic and expensive things to try to help them. What we really ought to do is identify people with a predisposition to disease and find some kind of effective treatment that will stop it from happening. And that's not a new concept. So here is a slide showing a patient on the left with phenylketonuria. So this is an inherited disorder of metabolism. The patient lacks phenylalanine hydroxylase, so they accumulate very high levels of the amino acid phenylalanine in their blood and urine. And if that's not identified early, the results are catastrophic. You'll lose 70 IQ points in the first year of life if you're not on the diet. So that was a big problem, and that's why that was the first disease for which there was newborn testing. So that was instituted in the 50s. I was born in 1954. I don't see too many people here who are likely to be born before that. So most of you were probably tested for this disease. And it makes sense because it doesn't cost very much to take a drop of dried blood from the heel and test everybody to identify the small percentage of people who have this disease, because the payoff for intervening with the diet is so huge that it justifies low-cost screening of the entire population. So that's one of the concepts of precision medicine. But of course, in the mid-50s, we didn't have genomics. It's now done by mass spec. It's not done by sequencing DNA. But when it was first introduced, it's the Guthrie test. That's the physician who invented it. And he had a strain of bacteria that's a phenylalanine auxotroph. It requires phenylalanine to grow. It can't synthesize its own. So you put dried blood spots on a plate that's been spread with this strain, and you see how big the colony is. And if it's little, you're normal, and if it's big, you have PKU. And that's how it was done. So precision medicine doesn't require genomics, but it's going to be greatly advanced. So thinking about tests that we really ought to do that have a big payoff, there's a lot on that slide. Let's not worry about the details. This was a set of recommendations from the American College of Medical Genomics in 2013. So they tried to be non-controversial, and they were non-successful. But they tried to come up with a list of genetic variants that are so awful that you'd want to inform people of these as incidental findings if you sequence their genome for some other reason. And so these include things like BRCA, which I just mentioned, and also some other cancer susceptibility genes. But here's an example of another variant that is on their short list. These are several genetic variants that contribute to hypertrophic cardiomyopathy. So the heart on the right has an overgrowth of muscle, and you'd think more heart muscle might be a good thing, but if you notice, the chambers inside the heart are a lot smaller, so total cardiac output is lower. Unfortunately, many of these people are completely asymptomatic, and the presenting symptom is death in the second decade. So you would really like to identify these people in advance. There's corrective surgery, or just implanting a pacemaker can add years to their life. So a cheap screen, if it were cheap, and an inexpensive medical intervention, implanting a pacemaker, gives them many extra years of life. So that's a payoff at some point if sequencing genomes is cheap enough. So there are a couple areas of medicine that are impacted by genome sequencing. So we've talked about inherited disease, so the cancer susceptibilities, the newborn stuff. That's one aspect of it. The big application of genomics in medical care now is in cancer, because cancer is ultimately a genetic disease. Cancer results from the accumulation of somatic mutations in genes that affect cell growth and division. And if you get enough of the wrong kind of mutations, that cell becomes cancerous. So the application of genomics in cancer, besides as a research problem, is in diagnosis. So a pathologist might look at two tumors that are absolutely identical by pathological criteria, but they're genetically distinct, and they would respond to targeted drug treatment very differently. So some anti-cancer drugs that are highly specific for particular mutations will be highly effective against some tumors, but will make others worse. So genomics is becoming a routine part of cancer care. We're not all the way along, but we are heading for a future where specific drugs that target specific genes will be the norm in cancer care, and chemotherapy and radiation, which are general purpose cell killing techniques, will vanish. That's the goal. And of course, there's pharmacogenomics. So our capacity to metabolize drugs or to respond to them is in part due to our genetic makeup. So there are some people who should never take drugs of a particular kind that are safe for everybody else. And for other drugs, everybody needs their dose adjusted up or down depending on their genotype. So pharmacogenomics is also an emerging area. So these three areas now are about a $13 billion industry right now, and heading to about $22 billion in 2020. When you do company pitches, you always have to include those numbers. Okay, so let's talk about technological change. DNA sequencing technology has changed extremely rapidly in my career, and I'd like to show you the slide that everyone who has ever been funded by NHGRI is required to show. And that's this one. You may have seen this before. This is the cost of sequencing a human genome year by year. And if at the completion of the Human Genome Project in 2001, you said, hi, I'm fabulously wealthy, I'd like to have my genome sequenced, and I'll write the check, it would have been $100 million. $100 million to sequence your genome in 2001. I'm guessing that's not when you guys got yours done. So then you see it collapses, and in 2014, we sort of hit the long-standing milestone of the $1,000 genome. So that's 5 orders of magnitude in 13 years. The top line, oh, that's a semi-log plot in case you hadn't figured that out. The top line is Moore's law. So that's that spectacular doubling of transistor density every 18 months that has made the semiconductor industry so interesting. So how do we know what's going to happen next in DNA sequencing technology? I'd like to look at the information processing industry as well. All right, and so these aren't my slides, they're Alan Kerr's ones. So we're familiar with the concept of linear versus exponential growth. So if I take a linear journey, each step is a step, and each step puts me one step ahead. If I take an exponential journey, every step I take is double the size of the last step I took, and it really adds up. So for some reason, the concept of exponential growth and the implications, it tends to elude our imagination. So even people working on the Human Genome Project, critics of the Human Genome Project said, look, you're 7.5 years through the project, and you haven't even sequenced half the human genome yet. You're going to fail. And they said, no, we're ahead of time and under budget. And that's exactly what happened, right? So that's the implication of exponential growth in the sequencing field. So here are the data for transistor density. So these are microprocessor transistor counts over 4 decades on a semi-log plot. And it's like clockwork, right? So this is where Moore's law comes from. So it's not just transistor density that's changed that way. This is processor performance in millions of instructions per second over a similar time frame, and you see it's also exponential. Similarly, the price of an individual transistor falls exponentially over time. That's not surprising, all of those things are related. But what's interesting is here is the cost, bits per dollar of magnetic data storage. Now that's not directly dependent on transistor density, but this technology has also moved in an exponential way across the same time frame. So let's go back a bit. This is a picture of ENIAC, sort of one of the world's first large computers built in 1948. And just as a measure of its performance, it could do 5 times 10 to the 3rd floating point operations per second. Can anyone tell me what's missing from that picture besides people? What's missing? Keyboard, that's good. If we were to open the machine, what's missing? Transistors are missing, right? Because transistors were invented in 1947. They weren't commercially available in 1948. What's on the inside of ENIAC? Vacuum tubes, right? Okay, so here, keep that in mind. Here is a modern, well, the slide might be 3 years old, supercomputer that delivers 10 times 10 to the 10th floating point operations per second, so 12 orders of magnitude faster than ENIAC, right? Now, the reason I put ENIAC up there is this. The exponential growth of processing power. This is how much processing power you get for $1,000, right? Using over time. And this is over 110 years. So there's exponential growth over 5 different technologies. The Jacquard loom, so electromechanical stuff, relays, vacuum tubes, individual transistors, and integrated circuits. The technology just plows on exponentially growing, even though the underlying technology is changing. So you saw that in the case of magnetic storage. You see ENIAC is just a blip on here as we transition right through vacuum tubes and into something else. So I don't know whether that's just because we've stepped back and we're looking at 110 years or not, but I think it's generally true that we're going to see exponential growth of technology no matter what the underlying technology is. So, of course, one technology tends to substitute for another as we iterate through the development of devices. So there's really kind of an S-shaped curve for the adoption of any technology. There's a time when we're trying to get it to work, and there's a time when we're really improving it, and then it kind of plateaus out as we squeeze the last bits out of it. So I'm not really a semiconductor guy, and I don't know where we are on the semiconductor plot, but I know that we are a lot closer to the theoretical limits of what semiconductors can do than we were when I was a kid, right? So we're going to be in the point where we're squeezing the last out of semiconductor. I don't know what the next technology is, maybe some of you do, but the prior slide suggests that we're just going to blow right through that theoretical limit, and we're going to move to another platform, and we're going to keep seeing exponential growth. So with that in mind, let's look at this. Now, this is over a much shorter time period. Obviously, something happened in the mid-2000s that collapsed the price of DNA sequencing. But, you know, the $1,000 genome, which was this elusive goal for so long, you know, we kind of achieved that 2 years ago. But we're buying genome sequences right now, and we're not getting them for $1,000. So, you know, have we squeezed the last that we can out of the existing technology, or is there still more to be had? So, can we have incremental improvement, or do we need a revolution? And unfortunately, I'm put in the very peculiar position of being an incrementalist. It's a lot more romantic to be a revolutionary, but I think we can take what we have right now and tweak it and put it together in new ways and push, you know, through that last barrier. So what's that last barrier? You know, why isn't $1,000 good enough? So this is actually about the wrong question. The question is not how much does it cost to sequence a human genome and how proud we are of how much progress we've made. The question is, what is the medical value of a human genome compared to its cost? So when I asked how many people had their genomes sequenced, not very many hands go up. So obviously the medical value of a genome right now is quite a bit less than its cost, or more people would have been done, right? And you guys are early adopters like me, right? So how do we estimate what the medical value of a human genome sequence would be? So if we take the ACMG list, the things you really want to report as incidental findings because they're highly actionable, and we take the ones that are most prevalent in the population. So the top 7 of the 60 or so account for about over 90% of the incidence of those things in the population. If we just identified those and estimate what the value in quality adjusted years of life added would be over the whole population, we get a value for sequencing people's genomes. Now this is nothing but those 7 genes. And what is that number? The number is $500. So as soon as you can sequence a genome for $500, and that means, you know, the person that talks to the person, the storage of the data, you know, all that, the billing, all that stuff. As soon as that's $500 or less, it pays to sequence everybody. Most people are going to be fine. A small percentage of the population will be identified as having a highly actionable variant, where if you don't intervene, there are expensive consequences. And that justifies sequencing everybody. So that's only considering those 7 genes in healthy 25-year-olds. What if we sequence newborns? Then the value of the human genome goes up, right? Because now we're getting all the newborn things and the things that tend to kill people by the time they're 2 years old. What if medical knowledge increases, as it will? Then we understand the effects of more and more variants. So the value, the medical value of a human genome will only rise over time. And the cost is either going to stay the same or go down. We hope it's going to go down a lot. So let's talk now about DNA sequencing. And I've heard that lots of people here use genomic sequencing, but they're not working at it at the level of chemistry and not thinking about how it works. They're used to just getting the hard drives in the mail the way we do. So that's what DNA looks like. It is a double helix, and the 2 strands are held together by weak hydrogen bonds, base pairing between A and T and C and G. And that means if you separate those 2 strands, all of the information necessary to build a molecule is present in the half strand, right? Now, you don't need 2 strands to have all the information because they're complementary. That's very cool. That's a polymer of individual units, so that's a deoxyribonucleotide. There's a sugar that has the blue shade on it that's deoxyribose. It has 1 to 3 phosphates on it, and then it has a nitrogenous base. So the phosphate is on what's called a 5' carbon, and notice that there's a 3' hydroxyl. Those 2 are the ones you got to watch. So the primary structure of a single strand of DNA, the 5' phosphate is joined to the 3' hydroxyl of the adjacent base in a phosphodiester bond. So any one strand has a 5' end and a 3' end. And then base pairing is hydrogen bonds between A and T or between C and G. So the final detailed look at the structure, the two strands of the double helix are anti-parallel. One goes 5' to 3', the other goes the other way, 3' to 5'. And those things are all important. And there's the final molecule again. So when DNA replicates, it does so in a peculiar way. Fortunately for us, it is perfectly happy to do that outside of the test tube, or outside of the cell and in a test tube, being supplied with only the enzyme that does that, some cofactors, and deoxyribonucleotides. It's very easy to replicate DNA outside a cell, which is great, because that's how DNA sequencing is done. So think about when DNA replicates, the peculiar requirement is, of course, you need a template strand. That should be clear. But you also need a primer. You need a little bit of double-stranded stuff. And in normal DNA replication, that would be RNA. In contrived situations, that's perfectly fine. You have DNA. And notice that the 3' hydroxyl is available. And then you import a new base by base pairing. So the enzyme checks to see that that's the right base. And then it cleaves that phosphate for energy and forms the phosphodiester bond. So that's how it works. But DNA can only grow from the 5' to the 3' direction. So now that you have been caught up to speed on DNA replication, you're in a position to understand one of the earliest DNA sequencing techniques that's still in use today, and that's the basis for the major market player. And that's Sanger sequencing. So here are two sugars now. One is deoxyribose, which we've been talking about, and the other is dideoxyribose. So deoxyribose is missing the hydroxyl from the 2' carbon, but dideoxy is also missing the 3' hydroxyl. So what would happen if we were to poison a DNA sequencing reaction, or a DNA synthesis, with a dideoxy base? What would happen is that base would be added, but since it lacks the 3' hydroxyl, that strand can no longer be elongated. That is the end of the line. There is nothing to form a phosphodiester bond with the incoming 5' end of the next base. Right? So that's the basis of Sanger sequencing. Imagine that we have a template strand that we want to sequence, and we have made a specific primer to that, either priming to a cloning site, which is the way we used to do it, or just PCR primers or whatever. And that's a prime template. So we're ready to extend that top strand from the 5' to the 3' direction, base pairing with the bottom strand. Now imagine that we split a bunch of those into 4 tubes. So there's the prime DNA template, we have DNA polymerase, and we have DNTPs, so deoxyribonucleotides. And now imagine that we poison each of those reactions with a dideoxy version of A, T, C, or G, and attached to those nucleotides is a fluorescent dye. So let's look in the A tube. Here's the primed template, and as we allow synthesis to proceed, maybe about 1% of the bases, or the DNTPs are dideoxy NTPs. So if they're inserted, they'll stop template growth, or base addition. And so we'll end up with a collection of molecules, all ending with dideoxy A, but they all end with A. Right? And the same thing is going on in all 4 tubes. Now, if we take all of those molecules and melt them, so we're just looking at the newly synthesized strands that end in fluorescent molecules, they will differ in size from each other by 1 base. We could run them on a gel or a capillary column and just read from smallest to largest. We're actually reading the sequence because we synthesized it, right? So A's are going to be green and whatever, C is red or whatever. And we can just read the sequence once we separate these fragments by size. And that's how Sanger sequencing works. And just to show I'm not making it up, here's an actual picture of a lot of Sanger sequence data. And fortunately, this is all done by machine now. So, how can we take that and the existing parts and do this incrementalist thing of pushing DNA sequence to be cheaper than it currently is? So let's talk about some cheap ways to look at genomes, some things maybe less than whole genome sequencing. So there's 4 things I want to mention. PCR, oligonucleotide arrays, exome capture, and of course, whole genome sequences comparison. So PCR is polymerase chain reaction, and I guess if you watch crime shows, you've heard of this. So the origin of this is actually our national parks. There's a bacterium that lives in boiling hot springs, and it naturally has a DNA polymerase that is not heat sensitive. So the ones that we have, if you heat it to 95 degrees, it'll denature just like an egg white in a boiled egg, and it won't be DNA polymerase anymore. But the bacteria that live in these boiling hot springs have a thermal resistant DNA polymerase, which is really important. So imagine we throw genomic DNA, and we're going to show you a very small piece of the genome here, in a test tube, and we melt it, right, by heating. And if we have the heat-resistant DNA polymerase and DNTPs, we're good to go. The only thing we need is primers. So we pick two very specific primers to a sequence we're interested in and add them in vast molar excess. So when the strands anneal, the first thing they're going to do is anneal to the primers. Then we drop the temperature to allow DNA synthesis, and those primers will extend. Right? So we can melt it again for another round. And if we do that through 20 or 30 cycles, those things defined by the primer sequences predominate until in the end, you wouldn't even see the genomic DNA that was the import. After 20 or 30 cycles, the only thing present in any appreciable quantity is this sequence that we've amplified by putting primers to the specific ends. So the way that's used clinically, you can either score, what size of fragment did we amplify, and that's what's done in forensic testing, or you can just take those amplified segments and load them straight onto a cheap sequencing machine and sequence them. And that's what's done in the cancer and pharmacogenomics panels. Right, so we'll come back to where that fits in the scheme. So now let's talk about DNA hybridization arrays. Has anyone ever done 23andMe? Yeah, a couple of early adopters. Okay, so here's how it works. It's a hybridization array. They're not sequencing yet. So you spin the tube. You probably have heard that part. And then what they're using is a hybridization array. So this is an Affymetrix gene chip. And what it has on it, this is pretty remarkable technological development. We figured out in the 90s, I guess, how to print oligonucleotides on silicon using photolithography. So there's a very complicated mask that goes into the making of these things, and there's 2.4 million spots, each having thousands of copies of a specific oligonucleotide. And so each spot has thousands of copies of an identical sequence. And when DNA is hybridized to those, you can pick hybridization conditions that require an exact match. And that's important because the base shown in red here is a known site of variation in the human genome. So that's a base that can be present in one flavor or the other flavor in the population, and the rest of those bases don't vary. You can set up the hybridization conditions so that only an exact match will hybridize. And you can set that up. You make 4 spots, each with a different base in that position, and you're prepared to score that variant. So your DNA is sheared and melted and biotinylated, and then it's hybridized to that array. And if an oligonucleotide array spot hybridizes to your DNA, now there's a lot of biotin over the top of that spot. And you can take fluorescently tagged streptavidin, which is a biotin scavenging protein, bind it, and now you've lit up the spot. So remember, we have all 4 flavors of any possible site of variation. And so at most, from any one person, there will be 2 spots lit because you can be heterozygous, but you can't be trisomic. And an image of the chip is shown there, and you can see why we pay computers to decode that rather than interns. And that's how you get 600,000 assay results from the 23andMe chip. Okay, so I'm going to briefly mention exome capture because I think it's on the way out. So I hope this isn't news to any of you. Only about 2% of your genome encodes protein. The rest of it, well, I mean, I won't call it junk because I don't want to slander our ancestors, but that's basically what it is. So 2% is protein coding. That's where all the action is in terms of looking for genetic variants that might impact health. So why don't we just capture that 2%? Sequencing 2% is cheaper than sequencing 100%. So that's been done. And then, of course, whole genome sequencing is the gold standard. So how do these stack up in terms of cost and capability? PCR is great. PCR costs us like 20 bucks a thing that we want to do. We're going to be selling PCR tests on horses. All of the cancer panel stuff and pharmacogenomic stuff, those are PCR tests. They're wonderful. The drawback is it doesn't scale. Every time you add a site, you add a unit of cost, right? There's no way to drop the cost of PCR by 5 orders of magnitude, because you need your primers and your polymerase and, you know, in some way your sequence or size fraction of the variants. So that's not going to be subject to a lot of improvement. It's extremely incremental improvement. Nothing's going to be order of magnitude. Arrays are fabulous because they really scale. You know, 600,000 sites of human variation scored for $200. That's great, okay? But what's the drawback there? That only scores sites of variation that we know about. So I don't know how good a look you got at your sequence or what kind of data you got back, but when I was sequenced, I had 4,000 or 5,000 private variants that were not in databases, that were in coding sequences, right? And as more people are sequenced, that's dropped. The Precision Medicine Initiative is going to sequence 1 million people. So the number of private variants that any newly sequenced person is going to have is going to go down, but it's not going to go to 0, because they're still very rare, either because they just arose or because they're selected against variants. So everyone is always going to have private variants. If you use an array, you can only score things you've already seen. So arrays are great for what they do, but it stops at finding novel variations. And certainly for sequencing cancer genomes, you want to see exactly what the mutation is in each oncogene that's been mutated. You do know, yeah.
So what's the cost of rerunning the array?
You can't. It's one
It's
shot.
one time
Razors and blades. Great business to be in, terrible business to be on the receiving end of. Right? Great question. All right, exome cost. When I actually had my exome done in an experimental program at 23andMe, it cost me $1,000. So I'm figuring it cost them $500. These are a little bit wonky. There's different kits from different manufacturers. They capture exomes, you know, more or less well, but each kit differs from the other kit. This is never going to be great, and all we have to do is crush the cost of sequence of genome, and exome capture will go away because it's messy. So whole genome sequencing now is about $1,000, and we have to push it down well below 500 in order to get precision medicine. So how's that happening? Well, here's a picture of who's playing in the market. Illumina is the 800-pound gorilla. They use a method of sequencing that's like Sanger sequencing. It has improved chemistry and vastly improved instrumentation, and they are delivering basically the $1,000 genome right now. So LifeTech is the other player. We haven't talked about how that works, but we're about to, because that's the area in which ElectroSeq initiated its research efforts. So we were first focused on improving semiconductor-based sequencing, which is the LifeTech method. So now I'm going to tell you a little bit about some R&D we did at ElectroSeq, and that is a sort of science fair project version of our fluidics setup, but it works. There's relays and an Arduino board in there, so it's the real thing. Okay, so semiconductor sequencing is also sequencing by synthesis. So in order to determine the sequence of a DNA molecule, you synthesize the strand. All right? So it starts with primers and templates, just like always. But in this case, the deoxyribonucleotides are not there. We have the prime template, we have the DNA polymerase, we have the cofactors, and we set everybody up and ask them to wait. And what they're waiting for is a base. So we give the bases one at a time. If we give it a C, nothing's going to happen here. This one's waiting for an A. If you're ahead of me, you'll see that's a D, and so it's waiting for an A. So it won't do anything. C will not be incorporated. If we wash out the C and then add A, the A will be incorporated. And when the A is incorporated, it does a number of things when that phosphodiester bond forms. It kicks off pyrophosphate, which was the basis for a now obsolete method, the 454 method, that detected pyrophosphate. It also throws off a proton. So all we need to sequence DNA is to do DNA synthesis reactions with normal bases, not chemically modified bases, in a test tube, and have some mechanism for detecting protons. And the instrument that does that is called a pH meter, right? So if you just added bases one at a time, you're asking a series of yes or no questions. Did you add a base? Yes or no. Did you add this base? Yes or no. And you record all of those answers, and you will derive the sequence from synthesizing the new molecule. So imagine that we have those test tubes, and that the semiconductor technology makes it easier to make very small test tubes and very small pH meters. So each one of those wells, we're showing 16 here, has a primed template that's waiting for its next base. So we flood this with an A, And all of the wells that were waiting for an A light up their pH meters because they throw off a lot of protons. We wash that out and add a T, a different set of wells light up. Same thing C, same thing G. And each of those wells is reporting yes or no, yes or no. Did you add a base? Did you not add a base? And we know which base was on top at the time. So we can read the sequence out from any number of wells by flooding them with that, with just a series of different bases and recording the answers. So that's how the LifeTech ion torrent and ion proton work. And here is our incremental innovation. So what's in those wells is not actually a pH meter. What it is the DNA sample is like that pie that's on the top, and the bottom of the well is tantalum oxide, which interacts with protons, and beneath that is a semiconductor, so a transistor. So it's an ion-sensitive field effect transistor. And the difference between the ion torrent design and ours is that we're using a dual-gated ion-sensitive field effect transistor like that found in flash memory. So the way this works, if you put a DC voltage on the floating gate, that sensor operates in what's called avalanche mode. So a triggering event causes a cascade of events in the semiconductor, shown in the bottom part, that amplifies the signal about 7-fold. So a floating gate sensor with a DC voltage loaded on the floating gate is more sensitive than the other kind, and you also don't have the area overhead required from having a reference sensor. So it's more sensitive, and it doesn't eat up as much space on the chip. So how's that high sensitivity do us any good? You can't detect proton emission from a base addition from a single molecule. You have to amplify the molecules in order to sequence them. And the way that LifeTech does this, they do it by PCR on a bead, and they end up with a bead that's about 3,000 microns in diameter that has 800,000 copies of the sequence that's to be sequenced. Because our sensors are more sensitive and smaller, we have tried to get away with what's called Rowley amplification to make a nanoball of single-stranded DNA that has a lot of self-complementarity that has only 5,000 copies. So if our detection was 160 times better than LifeTech, it would work. So the first prototype chip had 35,000 sensors, and there's 4 different architectures here. There's N and P, and then 2 different sizes. Here's scanning EM of the wells, and here's the wire bonded chip, and then a crude fluidics cap, which we have since improved. But you wouldn't be seeing all this if we didn't have actual results. So one of the early runs, we have DNA synthesized by Rowley amplification of known sequence in all of the wells. When we add the incorrect base, which is T shown in black on the top, there's not a big signal. When we add the correct base, which is G, there's a massive signal. So I know this doesn't look like much, but it's like when people show you, the sonograms of the baby. When, we have detected the proton signal from Basic Corporation from 5,000 copies instead of from 800,000 copies. So this says that this is going to work, and it's just engineering from there on. So what's the goal with that? The goal was to build a chip that has 1 billion sensors, and that would be capable of sequencing 10 human genomes in parallel at a cost of $100 per genome. And that still remains a long-term goal for ElectroSeq, although we have adopted another project that has gotten us a little further along in the interim. I'd like you to meet our team. Payman Zarkesh-Han, who's an expert at systems, sensors on a chip, and he's in the electrical and computer engineering department. Jeremy Edwards, who has been involved in improving DNA sequencing technology for the past 15 years, and then my background is in genomics and bioinformatics. So I've mentioned 3 things: precision medicine and the promise of precision medicine, and that's something we can all get on top of, because there's other aspects to it besides genomics. There's also a big data aspect and a lot of public policy aspects. We talked about technological change. I think if any of that is new to you, I think you can carry away from that. There's kind of a general way at looking at technological advances and what capabilities we're going to have in the future at kind of a predictable interval, even not knowing what the underlying technology is. And finally, I've talked about how we're going to make some incremental changes to DNA sequencing to push us past the point where we will invert medicine. We will get the cost of sequencing a human genome way down below what the value is, and everybody in here will be sequenced as a routine part of standard medical care. So those are the bits and pieces that we will use to tinker incrementally with the process of DNA sequencing. I've done a lot of pitching to investors. You always have to show a slide like this, so I thought I'd show you what one looks like. Phase 1, we're doing R&D. And phase 2, we are building perhaps an advanced prototype that could be manufactured at scale. We have 2 possible exits. One is to be acquired for IP at the end of a demonstration phase. The other is to be acquired for quite a bit more after we have a prototype that could be built at scale. And again, I wouldn't be showing you that slide unless there were a good story to go with it. And the story is that in the past several weeks, we have gotten to the handshake stage of a deal of a funding round that's serious. And I'm not going to tell you the details, but you'll be able to hear about it in the press in the next few weeks. And we are definitely going to be on track. So every pitch ends with an ask, and here's my ask. That's my contact information. If you'd like to talk about any of the things that I talked about, or you think you have something to offer at ElectroSeq, please look me up. They've got the slide set, so you don't even have to write down the contact information. And I'd be glad to hear from you. So, glad to take any questions too. Yep.
So are you working with individual customers or with companies?
This is just the technology development phase. So the thing that's made it really difficult to talk to the rest of the entrepreneurial community about this company is that we have no products, services, or customers, and we will never sell anything to anyone except the company, right? That's the model. So we strictly generate IP and a working prototype, and then we'll be acquired. So there will never be any direct contact with the public. The big players now, I mean, and why am I smiling when I say that, right? Because if you want to deal with human, which is essentially 100% of the market, you've got the FDA, you've got Medicare reimbursement, you have all these things that I am not interested in, right? So one way to build a company is to develop IP, and then when it's far enough along, the big boys say, you guys can't play with this anymore, you're fired as CEO, which they've already discussed with me, and then we're acquiring your company. That's great, that's how it works. So any of you who are involved in tech development, that is one path. And I think it's a really different set of skills to be talking about negotiating the FDA and handling Medicare. You're shaking your head knowingly, right?
Well, I was just thinking about all of the ethical considerations and the policy considerations.
Yes, well, yeah, well, so ethical stuff, 3% or something like that of the Human Genome Project went to ethical, legal, and social implications. Of course, this sort of quadrupled that field, right? Because 3% of the human genome, I mean, that was $3 billion back when $3 billion was real money, right? So those are massive problems, and we're not involved in any of those problems. You probably know about 23andMe, right? Right. So they got told you can't hand this information out to people. So I'm sort of an information libertarian. It's like you ought to be able to go into the hardware store and buy a DNA sequencing machine and sequence yourself if that's what you want to do. That's just me. So I don't do public policy. Anybody else? Yep.
I think there's a good comparison with the Illumina techniques, which is basically going to the market. Everybody uses Illumina now.
Yes.
So I wonder, I'm not clearly the background, I mean, on this molecular level, amplifier. So what is it exactly, or talking about the difference between your technique and the Illumina technique? And how do you say that their market right now is a...
Yeah, they are the 800-pound gorilla, yes.
Yeah, they were probably improved, and then how do you compare, you were talking about not right now, probably a couple years,
yep.
the new technique in the
Right.
drug, they were also improved. How do you
Yeah.
say that?
So first let me say that I can't completely answer your question because I'm on video. And so we have proprietary things going on that give me the confidence to stand here and say I'm not worried about it. The second thing to say is, let's actually look at what's happening in an Illumina machine. Okay, so it's not test tubes, there's spots, in an array, and there's amplification of those spots. So there's a flow cell, and they're running all the reagents past these spots, and then watching them turn colors, right? Until extremely recently, you still can't get the machine. Inside the machine, is a microscope with a camera taking pictures of the flow cell, right? Come on, right? What year is it, right? So that's got to go, so they can improve that. Is there a limit to how small they can make their wells? Yes. Yes, there's a limit to how small their little sample colonies can be because they're detecting it by fluorescence. So there is some theoretical limit, which is too few photons for whatever they're using to detect it. So can Illumina improve its system by 5 orders of magnitude in the next 3 years? No, they can't, right? They're going to have to go to a new technology. Okay, that's my opinion. Can we look at anything Illumina's done to say that there might be somebody at Illumina that thinks that might be true? Okay, does anybody know about the X10? Right? You guys just send stuff off and get it sequenced, who cares, right? So a couple of years ago, they released the monster of Illumina sequencers. It's the Illumina X10, very high throughput, and they cost $1 million each, but you can't buy one, you have to buy 10. Right? So you have to invest $10 million plus a $50 to $75 million wrapper around it. There's, I think, 21 of those sites in the United States. Right? Now they can do a $1,000 genome if it runs 24/7, 365. Right? So why did they do that? They did that because they knew that the spreadsheet guys were never going to let the people who bought that put it on the loading dock for eBay after 2 years. That's going to have to be amortized over 5 or 10 years. So that machine is going to keep operating until it's paid off, right? So why would they do something like that? Why didn't they go for the low end? Because they know they've squeezed what they can out of their chemistry. They had to go with high throughput instruments that put a financial constraint on the people that bought, right? Now, I don't know that from Illumina, but if they ever see this tape, I mean, I love what they do. but I mean, they know that they can't improve their chemistry by 5 orders of magnitude. So they have to do something else, right? So there is going to be some version of semiconductor detection, so no more cameras, right? And no more gels, and there's going to be some version of targeted sequencing. So not sequencing the 98% of the genome that we don't care about. So those are the incremental things. What we haven't talked about are the revolutionary things. So I suppose you've probably heard of nanopore stuff. That is a lot further down the road. It might not ever appear. So it might be like gallium arsenide semiconductors, the material of the future, and it always will be, right? So maybe it'll work, maybe it won't. There will be something revolutionary 10 years from now. I don't know what that is, but that's not of any relevance to ElectroSeq. Yep.
Why is array sequencing a one-shot deal?
It's not sequencing. It's array hybridization.
Array hybridization.
Because you just put, you just washed gunk overall. How are you going to get the DNA off? What's the DNA stuck to? It's stuck to other DNA. So why don't we just DNase the whole thing? Oops, the array went away. I mean, you can't just melt it off and be guaranteed that you're going to get it all off. So the only way to get a clean signal is to use a fresh chip every time. And they're $200 a shot, and 23andMe sells them for $99. How do they do it? Volume. No, they're going to monetize the data. Right. So that's my favorite story. Is how they're doing it. Yeah. So there are sequencing technologies we're looking at, like the ion torrent chip, and our chip is one use only. And that makes us really happy because we could sell the machines at cost or below and make all the money on the chips if we were in the market for selling. So those are very attractive business models, razors and blades. You practically give away the razors and you charge premium what the market will bear for the blades. No one's asked about the informatics end of it, right? Not really an important question. So somebody asked me when I was here a month or so ago about that, and I said, the informatics software does not know the means that were used to derive that sequence. It already is in place to analyze sequence once it's sequenced. It doesn't care how the sequence was generated. So that's the short answer to the question that I asked.
So what's the time frame for running one sample?
On a semiconductor platform, sample preparation, the Rowley amplification might take a day. Running a complete human genome on one chip would take less than a day. So the Illumina machines now are like 12 days. So there's a giant backlog to get on them. So the idea would be what the market needs is a cheap machine that can be operated by semi-skilled labor, right, in a clinical setting that has a user interface that's friendly. So your whole genome is sequenced, but your physician is only interested in the pharmacogenomics part, and they just click the selector to pharmacogenomics. They get the executive summary, and they say fine, right? And they don't keep your genome sequence. That might deal with the privacy issue, right? So, hey, it's fine with ElectroSeq if people want to just buy a new chip. Because, you know, once we have your genome sequence, we have it, right? So, but if it's $100 to get it, they're not even going to store it. And they'll just buy more chips. So that's good news for us. Thanks for asking. Yeah, so all kinds of dimensions to this problem. So we've just talked about the technology part, but the public policy part is enormous. All right. Thank you very much.
Thank you.